Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn a detailed mechanism of uh, regeneration that is pertaining to the fin in zebrafish model. So we have seen how efficiently zebrafish is able to regenerate various organs, and coming to different organs, say heart, say brain, say retina, say spinal cord, say fin, they adopt a different strategy. So if you look further into the retina regeneration, zebrafish exhibit epimorphic regeneration, in which non-differentiated cell mass formed after amputation is able to fully regenerate the damaged part, and it can be, say, limb, heart muscle, etc., etc. Epimorphic regeneration, the backbone of epimorphic regeneration is it is capable of restoring the morphology as it was before the injury. In 2003, scientists from Sanger Institute, they compared around 26,000 zebrafish gene to that of human genome, and they reported that almost 70% of the human genes have ortholog in zebrafish. That is a very interesting, exciting news, simply because although zebrafish and human doesn't look the same, although both are vertebrate, at a genetic level there is a lot of genomic homology and synteny, which is an exciting feature zebrafish possess when it comes to regeneration study. So Aristotle, very old Greek philosopher, in his articles dedicated to natural history, mentioned that animal embryos regenerate more efficiently than adults. So he has observed. So interesting thing about regeneration biology is that it is one of the ancient biology branch of biology, because everybody knew about planetary regeneration some 250 years ago, and still it is an evolving area of research. That seems like a conundrum, but that is a fact. Although the key genes responsible for limb regeneration and inducing dedifferentiation, proliferation, and differentiation of somatic and stem cells are similar in anamniotes and amniotes, that is, the animals that have got amnion or those animals without amnion. So that is the amniotes and anamniotes. In mammals, these genes are not activated after amputation. So we should understand that some animals, despite having the ability to regenerate, and the genes responsible for regeneration exist, sometimes they are not turned on in time, so which can account for your lack of regeneration. The process of fin regeneration is complete in around 2 to 3 weeks, around 14 to 28 days it is completed, depending upon the water quality, temperature, diet, pathogenic organism infection into the wound site, and also the stocking density, how many fishes you are putting in one tank. So fin regeneration, when you look closely, zebrafish, the process of fin regeneration have 5 stages. First stage is the wound epidermis formation, that is formed by not by proliferation but by the migration of the cells from the surrounding, and the mesenchymal disorganization, blastema formation, and regenerative growth, and the final stage is termination. So bleeding is stopped soon after injury, amputation. The first danger signal that is initiated by reactive oxygen species, ROS species, which attract immune cells in the site of injury. So ROS is capable of bringing in the cells, especially the immune cells, that is necessary for the regeneration to kickstart. Neutrophils arrive within minutes of injury, and it causes blood clotting, remove antigen, debris, etc. And wound epithelium formation is the next stage that is occurring. And what happens is the migration of the cells that is coming from the rest of the body into the epidermal cap, apical epidermal cap, is one of the initial stage and a necessary stage required for the regeneration to happen. And one of the pro-inflammatory cytokine, interleukin 1b, that is induced in the site of injury, which causes the inflammation. And this also credit goes to the M1 like macrophages, that is capable of, like you can see different types of cells, epithelial cells, neutrophils, M1 like macrophages, and ROS. They are all present here in this injured, and also you can see the osteoblast in this injured area. there is a lot of congregation of these cells, but the pro-inflammatory environment is one of the initial stage that is necessary. So macrophage depletion, usually people have done different ways of analyzing and understanding the fin regeneration. So how it works is the macrophage depletion that has resulted in significant decrease of the caudal fin outgrowth. So we know M1 macrophages are necessary for the pro-inflammatory cytokine, but a significant decrease in the caudal fin outgrowth happens because of the macrophage depletion. And it also causes changes in the fin morphology, including areas of aberrant tissue growth, defects in bony ray patterning, and heterogenic bone calcification. So what you understand, if you deplete the macrophages, the pro-inflammatory environment will not be perfect enough so that the morphology, the epimorphic regeneration can be established. So the macrophages regulate tissue homeostasis, organ repair, and angiogenesis, and migrate to the wound and secrete growth factors and cytokines, mainly pro-inflammatory cytokines, that in turn activate the ECM, extracellular matrix production. ECM is a must component so that these newly formed blastoma cells can migrate. So ECM is must, which presents all tissues and which can regulate the wound healing, providing a patch to protect the damaged area. So the ECM production by the immune response cells, ECM production is important for the establishment of structural stability to the injured area. In contrast to zebrafish and other fully regenerating species, mammals form scar tissue after amputation, which is nothing but overproduction of ECM protein after amputation through the deposition of fibronectin and collagens, the major components of ECM, leading to fibrosis, which can hamper, because it is overdoing the ECM production, which can hamper the proper migration of the cells, but the structural stability is retained in the injured site. In zebrafish, when bleeding following caudal fin amputation is stopped, the amount of fibrin and fibronectin decreases, what causes to prevent the scarring. Otherwise, scarring can happen. Prevent the scarring, because when wound is closed, proteolytic enzymes such as matrix metalloproteinases, that's MMPs in short form, remodel the matrix protein and to protect the healing of that organ or tissue and migrating cells to replace the lost tissue. So, or in other words, you can tell you want to occupy a house or a room, but if that room is having too much of junk, unless you clean that junk, no one can get into. The room had got 200 chairs. How will you enter the room? So you have to clean it. So this cleaning help is done by the MMPs. Blocking MMPs negatively influence fin regeneration. MMP enzymes positively influence activation of chemokines. Chemokines are molecules that allow the cell migration. They normally help in the provide directionality to the cells that stimulate the migration of immune cells to the injury site. And it also helps in maintaining homeostasis and also control cell migration during embryogenesis and regeneration. So these are all the important roles played by the MMPs. So if you block MMPs, you don't get these features, hence you don't get the regeneration. Another pro-inflammatory cytokine such as interleukin 1b that is induced at the site of injury, triggering regeneration inducing genes at that particular damaged area. However, in the absence of macrophages, overexpression of interleukin 1b, we already saw if you get rid of macrophages, you don't get proper regeneration. And we also know the interleukins are produced by the macrophages. So you depleted the macrophages and you overexpress interleukin 1b, it causes the apoptosis of the regenerative tissues. So what you understand from here, the role of macrophage is not just to produce interleukins. Their role is interleukin production plus many more. That is why if you want a normal epimorphic regeneration, the macrophages and interleukins are must. Just supplementing interleukin is not a compensation for absence of macrophages. So another pro-inflammatory cytokine, which is TNF-alpha, it was identified as a crucial component of caudal fin regeneration. This TNF-alpha is synthesized by M1-like macrophages. There are two types of macrophages, M1 and M2. M1 always does pro-inflammatory cytokine, M2 macrophages always do anti-inflammatory cytokine, and they get recruited to an injury region and exhibits dual action of enhancing accumulation of macrophages and participating blastema formation, because they can produce more TNF alpha. TNF alpha means more M1 macrophages. More M1 macrophages means there is more pro-inflammatory cytokine. So it's a kind of a positive feedback cycle. Another molecule, hyaluronic acid, has been shown to be highly abundant in the ECM of the regenerating fin, extracellular matrix of the regenerating fin. The enzyme, the hyaluron synthase 3, it's also known as HAS3 gene, is overexpressed within 6 hours after the caudal fin amputation and increases its maximum concentration within 24 hours. So what you understand, the hyaluron synthase, hyaluronic acid production is necessary in the first few hours of amputation. Inhibition of hyaluronic acid synthesis during the first 24-hour post-amputation, you block it by whatsoever mechanism. You can block the HAS3 enzyme, produces the suppression of blastema formation and regeneration, whereas the inhibition at later stages, say post 24 hours, you are inhibiting it, it had no significant effect on the regeneration process. So the initial few steps, that is the production or synthesis of hyaluronic acid within first 24 hours, is very crucial for the blastema formation to initiate. Hyaluronic acid molecules activate signaling cascades required for blastema establishment through the induction of specific blastema specific markers. This is one of the assigned function of the hyaluronic acid. Within 1 to 3 hour post amputation, HPA means hours post amputation, the site is covered by a multi-layer wound epidermis that is migrating from the surrounding side tissue of the fin, formed by non-proliferating epithelial cells distally migrating into the site and creating an apical epidermal cap. So these cells migrate into the wound site and create a cap called apical epidermal cap, which works as a physical barrier between the body and the environment. Of course, it will protect from getting infection. And at around, this is happening within 1 to 3 hours only. At around 18 to 24 hours, the mesenchymal cells underlining the wound epidermis, apical epidermal cap below that, mainly osteoblasts and fibroblasts, lose their organization and turn into a highly proliferative whitish mass of cells, which is called blastema, that is formed by around 24 hours. You can see under the microscope that can be detected under the apical cap. That is apical ectodermal cap is there. Beneath that, you will get a blastema cell that is formed. Blastema, which has two main functions. One is to support the outgrowth and to form specific structural elements essential for fin restoration. And it's mainly regulated by beta-catenin signaling, sonic Hedgehog signaling, fibroblast growth factor signaling, bone morphogenetic protein signaling, activin, insulin-like growth factor, and notch and retinoic acid pathways contribute to the activation and maintenance of the blastema. So many signaling events undertake the establishment of blastema and the migration at later stage. If you look closely, the TGF-beta signaling is very interesting. It is a graphical illustration of TGF-beta signaling, which has got two phases. One is canonical, another is non-canonical. Left side is canonical, right side is non-canonical. In canonical TGF-beta signaling, the TGF-beta ligand binds to TGF-B R1 and R2. Two receptors are there, TGF-beta receptor 1 and receptor 2 receptors, resulting in their activation to form a heterotetrameric complex. That means it will form 4 receptors come together and it will form a heterotetrameric complex. So the intracellular events that includes SMAD2-3 are next activated by the phosphorylation. Because of the TGF ligand binding onto the receptor, as you can see here, it formed a heterotetrameric complex. This leads to the activation of the SMAD2-3. They are activated by phosphorylation initiated by TGF-B R1 of this heterotetrameric complex. P-SMAD2-3 form a complex with the SMAD4, another free moving molecule, SMAD2/3, when they get activated, they will recruit the SMAD4 together with certain transcription factors. They activate specific target genes that contain SMAD binding elements. In short form, it is called SBE. So in the DNA of those gene regulatory regions, if there is a SMAD binding region, this complex, this SMAD2/3, can go and bind onto them, and they usually have those genes are implicated in the growth inhibition, apoptosis, ECM synthesis, and immune response. Whereas in the non-canonical pathway, so that what we have seen is a canonical pathway where the SMAD genes bind onto the SBE and causes the gene expression. In non-canonical pathway, TGF-beta induces the expression of a molecule called TRAF6 and ubiquitinate, and it ubiquitinates that particular TRAF6 molecule at lysine 63 dependent manner, promoting its catalytic activity. What happens to the TRAF6 then activates another molecule called TACE and PSEN1, and these two molecules get activated, resulting in the proteolytic cleavage of the TGF-beta-R1, the same receptor which forms a heterotetrameric receptor, TGF-beta-R1, generating a soluble TGF-beta-R1 intracellular domain. So you end up getting a free moving domain of TGF-beta-R1, and this is, in short form, it is called a TGF-beta-R1 ICD. ICD means intracellular domain. The endosomal adapter protein called APPL1 and APPL2 and also various microtubules are required for the translocation of this intracellular domain, TGF-beta-R1 intracellular domain, to the nucleus. So the function of this intracellular domain is in the nucleus, where it contributes to the activation of specific target genes. Whereas the SMAD is not involved here, TGF-beta-R1 is directly involved, and other modes of non-canonical TGF-beta signaling pathways are also been discussed and described. So that is what you are seeing here. In A, in the left side, you are seeing the canonical, whereas here you are seeing the right side, you are seeing the non-canonical TGF-beta signaling. And there is another TGF-beta signaling. They can also induce EMT, epithelial to mesenchymal transition, through the activation of non-SMAD pathway. This is also TGF-beta signaling, but it's a non-SMAD pathway where they involve something called Rho-GTPase via the phosphorylation as a polarity protein, also known as PAR6, leading to cytoskeletal rearrangement. So this is also non-canonical TGF-beta signaling, but it's a non-SMAD pathway where you have got a PAR6 involvement of the PAR6 protein, and this leads to the rearrangement of the cytoskeletal protein, often causing a breakdown of epithelial cell junction. Because we should understand, for the blastema to form, epithelium to migrate, etc., you cannot have cells attached onto each other. You need to break that junction so that the cells can move across. So the TGF-beta signaling plays some unique roles in defining the regenerative capacity of the fin. Another signaling pathway which can be called a sibling signaling pathway called BMP signaling, because both BMP and the TGF-beta signaling make use of SMAD proteins. Let us see how BMP signaling contributes. You may remember BMP signaling also is important in determining the dorsal area or the dorsalization of the planary embryos. You may recollect in the previous classes we have studied. So the complex regulation of bone morphogenetic protein signaling is quite interesting when it comes to the regeneration of the fin tissue. BMPs are processed by a pro-protein peptidase to generate mature dimers that then bind to two copies of the type 1 and type 2 BMP receptors. Two types of receptors are there, type 1 and type 2, generating a heterohexameric complex. In the case of TGF-beta, we saw heterotetrameric. Here we have got 6 copies, hexameric complex. Binding of BMP homodimers to their cognate receptors lead to phosphorylation of type 1 receptor and also by type 2 receptor in the GS domain. This is what happens, which is a domain present in these receptors. Activated BMP receptors then phosphorylate some different kind of SMAD. They are SMAD 1, 5 and 8. Earlier in TGF-beta we saw SMAD 2 and 3, okay, and 4. Here it is 1, 5, 8, proteins which dimerase with SMAD4. SMAD4 is common for both TGF-beta and BMP signaling, because rather you can say they will compete for SMAD4 and accumulate in the nucleus, where they mediate changes in the BMP regulated gene expression. So BMP has got its own target genes, not that TGF-beta and BMP, they act onto the same set of genes, separate set of genes. Regulation of this pathway occurs extracellularly via the binding of extracellular antagonists such as gremlin, GREM1 in short form, and noggin. These are antagonists of BMP signal, noggin, or in the plasma membrane via the action of pseudoreceptors such as BMP and activin membrane bound inhibitor. In short form, it is called BAMB. In addition, inhibitory constraints or receptor mediated SMAD158 phosphorylation, they can occur via FK binding protein. These are all amino acid residues, phenylalanine and lysine. FK means FK binding protein 1,2, FKBP12, it's the name of that protein binding, and inhibitory SMAD6 binding. This will be triggered in, which is relieved by the action of a methyltransferase known as PRMT1 methyltransferase. Additional regulation of BMP signaling occurs via the cytoplasmic phosphatase and ubiquitin ligases such as SMURF, and via microRNA and methylation mediated pathways which can control the BMP mediated gene expression. That means there is a checks and balances throughout occurring. So there will be a push and there will be a drag, and again there will be a push, there will be a drag. So this is the mechanism through which various pathways that works. So whatever we discussed is listed here, like BMP, it's get activated and forms a dimer, and it can be influenced by its antagonist. If they are present, they will not get a chance to bind to the receptor. If antagonist doesn't trigger, then they will bind, and it will lead to the activation of either the receptor can get activated and can trigger CoSMAD4 and RSMAD. They can work onto the gene expression event in the nucleus, or they can also get involved by other pathways where RSMAD can be influenced by other complex such as SMAD7, SMURF, and phosphatases. They can influence this RSMAD's 158 availability. And other mechanism is once the BMP is bound onto its receptor, they can be influencing the expression of SMAD6, and which also will tweak the BMP signaling events. So what you should understand is the BMP comprises of a large subgroup of ligands from the transforming growth factor beta family. Upon ligand binding, the BMP receptor 1 dimer phosphorylates the BMP receptor 2 dimer and initiates its kinase activity, stimulating the action of various downstream mediators. The main targets of BMP receptor and the phosphorylation are mainly the SMAD proteins, which are regulators of fibrosis and that translocate from the cytoplasm to the nucleus, where they bind to the DNA at SMAD binding motifs of various DNA sequences of regulatory elements of various genes, causing tissue-specific gene expression. Different homologs of type 1 BMP receptors are expressed in almost all regenerating fin tissue types, because BMP signaling is one of the pivotal signaling for fin regeneration. So BMP is active in the distal blastema, wound epidermis, osteoblast, and blood vessels of the regenerating tissues. Initially, BMP is essential to remodel the plexus into blood vessels. Naturally, the newly formed blastema need nutrition, which comes from the angiogenesis, that is a blood vessel formation. BMP promotes the deposition of collagen fibers into the basement membrane while closing the wound, that induces the actinotrichia formation, which is a unique structural protein of the fins. So if you look further, what are the early steps of fin regeneration? After amputation, what happens? The wound is closed within 12 hours post-amputation by migrating epithelial cells which form a multi-layered epithelium called wound epidermis. Next, the blastema, which is shown in orange in color. Accumulation happens and by a proliferative undifferentiated cells, which we often call them as blastema cells. Under each ray, fin ray, we have seen the structure of the fin. It has got individual around 16 to 18 fin rays will be there per fin, which is made of hemi rays, which will look like a bracket, which is for the structural stability. And next, the blastema, which is given in orange in color. Accumulation causes a congregation of proliferative mass of cell and differentiated cells. They are formed atop of the amputated site below the epidermis. Each ray, it keeps allowing, because the epidermis is covering the whole wound and the blastema is forming mainly on top of individual rays, and between the rays, the so-called inter rays, you have mainly fibroblast tissue. They are not a true blastema tissues. They distally migrating cells. During regenerative outgrowth, the blastema cells proliferate and differentiate to replace the missing tissue. Like you can see here, MSXB, one gene which is a blastema marker. You can see blue color right below individual rays, but you can see between two blue, there is a white area where there is no MSXB expression. There is fibroblast. They are not true blastema, but they are just a mass of cells. So if we look further, a few more molecular events. Inhibition of SHH signaling in the injured tissue by cyclopamine leads to the suppression of Wnt, beta-catenin, FGF and retinoic acid genes and loss of regeneration. However, activation of Wnt pathway results in the restoration of the regenerative processes. At the level of blastema establishment, the canonical Wnt beta-catenin signaling pathway regulating the stem cell pluripotency is critical to the blastema formation, which in turn is essential for the fin ray development. So the Wnt beta-catenin pathway during the regeneration process is to establish the organizing centers of the blastema to indirectly regulate the activity of other signaling pathways such as FGF and BMP signaling, which control the cellular proliferation and differentiation, which we have seen it already during the buildup of the cell mass. And the super expression of Wnt beta-catenin inhibitor, that is DKK1, and suppressing the tumorigenesis and cell proliferation in a normal scenario, it causes the cessation of the blastema formation and regeneration. So a negative regulator of Wnt signaling, if you overexpress it, you don't get normal regeneration. Knocking out of DKK1 gene results in the impaired limb and brain development during embryogenesis in mice. Thus, the regular Wnt beta-catenin DKK1 interaction is required to initiate the regeneration successfully. And if you look further the downstream events, what you can see is FGF is expressed at the early stages of fin regeneration that controls the blastema formation, osteoblast dedifferentiation, proliferation, actinotrichia, and the fin restoration. Errors in regulating the FGF pathway results in the severe abnormalities such as tumors is often seen. Fibroblast growth factor receptors on which the fibroblast growth factor act significantly contribute to effective regeneration. FGFR is essential to maintaining subpopulation of epidermal and blastemal cells to initiate blastema proliferation. A mutant zebrafish of FGF20A gene was temporarily knocked down. Epithelial cells covered the injury site, but the site do not complete the regeneration. Wild type fish, but we do not bring up a blastema cell formation. The wound was healed entirely without restoring the missing fin. So wound can get healed, but regeneration will not happen if FGF20 is deleted. It was found that the notch is activated in response to caudal fin amputation, but is not involved in cell migration or dedifferentiation after the blastema is formed. And you also, one more thing I want to tell you, if you cut the blastema in two different pieces, one proximal and distal, so the proximal cut will have more regenerative ability than the distal cut. So the site specificity also get influenced by genes like LDH2A and FGF signaling. So we will study more about regeneration biology in the next class. Thank you.